Today, we're diving into a group of biological entities that challenge how we define life itself. Viruses are everywhere. They shape ecosystems, influence evolution, and have enormous impacts on human health, agriculture, and global society. Even though they are incredibly small and structurally simple, their effects are anything but simple. As we move through the topic, we'll look at why viruses are made of, how they replicate, and how they interact with their hosts. We'll also explore why some viral infections are mild while others are persistent or even deadly, and how tools like vaccines and antiviral drugs are designed to interrupt viral life cycles. By the end, the goal is not to just memorize facts, but to understand why viruses are so effective and why they continue to be one of the most fascinating and challenging subjects in biology. Viruses are strange little things, and I mean that in the most scientific way possible. They are not cells, which immediately puts them outside how we normally define life. They do not belong to any kingdom, and they cannot divide or carry out metabolism on their own. Instead, they survive by hijacking a host. Once inside, they take over the host cell's machinery and redirect it towards making more viral particles. What makes them especially important is how broad their reach is. They infect bacteria, plants, animals, and humans, which means they influence entire ecosystems, agriculture, and human health all at once. Viruses are very small, simple, and incredibly effective. This is one of those questions that sounds philosophical but actually gets at the core of biology. Viruses sit in an awkward gray zone between living and non living. Living organisms grow, carry out metabolism, and reproduce on their own. Viruses do none of that independently. They do replicate, but only when inside a host cell and using that cell's machinery. Outside of the host, they are completely inert. Another important distinction is that viruses are assembled rather than grown. Their parts are made separately inside the host cell and put together like molecular Lego sets. Because of these limitations, most biologists do not classify viruses as living enough, even though they clearly evolve and adapt over time. Viruses escape detection for long times because they are incredibly small. Even the best light microscopes are not powerful enough to see them. It wasn't until the development of the electron microscope in the 1930s that scientists could actually visualize virus particles, which we called virons. Early on, viruses were grouped mainly by how they looked, things like size and shape. As molecular biology advanced, classification became much more precise. Scientists began grouping viruses by whether they contain DNA or RNA, whether that genetic material is single or double stranded, and how they replicate once inside of a host cell. This shift towards molecular classification helped us better understand how viruses function and how they are related to one another. Once scientists realized viruses were everywhere, the obvious next question was, where did they come from? There are three main ideas, and none of them fully solve the mystery. The devolution hypothesis suggests that viruses started out as free living cells and gradually lost genes as they became more dependent on hosts. The escapist hypothesis takes the opposite approach and argues that viruses began as bits of genetic material that escaped from cells and gained the ability to move between hosts. The self-replication hypothesis proposed that viruses evolved from early self-replicating molecules, possibly even before modern cells were formed. Each hypothesis explains part of what we see, but none of them completely accounts for viral structures like complex capsids. The origin of viruses is still one of the biology's open questions, which makes it a greater reminder that science is always in progress. Unlike cells, viruses do not have membranes, organelles, or cytoplasm. A complete virus particle known as a viron is basically a genetic instruction set wrapped in a protein. At the center of the nucleic acid core, which contains the genetic information the virus needs to replicate. Surrounding that is a capsid, a protective protein shell of repeating subunits called capsomeres. Some viruses also have an envelope which is borrowed from the host cell's membrane and helps the virus enter new cells. Viral genomes are remarkably small and efficient, carrying out only the genes that absolutely need. Despite the simplicity, viral structures range from clean geometric shapes to surprisingly elaborate designs. One of the most important ways we classify viruses is by the type of genetic material they carry. DNA viruses rely heavily on the host cell's replication machinery, which tends to be accurate and stable. RNA viruses, on the other hand, have to bring their own enzymes to copy the genetic material because host cells cannot do this. Those enzymes make it more mistakes, which leads to frequent mutations. This high mutation rate helps RNA viruses adapt quickly to new hosts or immune defenses, but also makes them harder to control medically. This is why many RNA viruses evolve so rapidly compared to DNA viruses. Viruses cannot infect just any cell. They must first attach to a specific molecule on the host cell surface called receptors. Viral surface proteins, often glycoproteins, bind to these receptors much like a key fitting into a lock. What's important here is that these receptors normally have regular cellular functions. Viruses have simply evolved to take advantage of those. This receptor specifically explains why certain viruses infect specific species or even specific tissues within the body. If the receptor is not present, the virus cannot enter no matter how much exposure occurs. Some viruses are simple, but others are impressively complex. Bacteriophages are a great example of viral engineering. The T4 bacteriophage looks almost like a tiny lunar lander, and it has a head that stores DNA and tail fibers that recognize and bind to specific bacteria. Once attached, the tail sheath contracts and injects the viral DNA directly into the bacterial cell. The capsid stays outside, acting almost like a molecular syringe. This structure makes bacteriophages extremely efficient at infecting bacteria and explains why they are being studied for applications like phage therapy. Adenoviruses show what a more symmetrical virus looks like. They have an icosahedral capsid, meaning it has many flat triangular surfaces that form a stable shell. Extending from the capsid are spike proteins that help the virus attach to the host cell. These viruses carry double-stranded DNA and are commonly spread through respiratory droplets or contaminated surfaces. In humans, adenovirus can cause illnesses ranging from mild respiratory infections to eye infections like conjunctivitis. Their structure is a great example of how viruses balance simplicity with functional precision. HIV looks very different from non-enveloped viruses. Instead of a rigid protein shell on the outside, it has a lipid envelope that comes from the host cell's membrane. Embedded in that envelope are glycoproteins that allow HIV to bind very specifically to immune cells. Just beneath the envelopes are matrix proteins, which help stabilize the virus and maintain its structure. Inside the virus carries RNA rather than DNA. This combination of an envelope, RNA genome, and specialized proteins is what allows HIV to enter host cells, replicate efficiently, and persist in the body over long periods of time. To make sense of viral diversity, scientists rely on classification systems based on structure and genetics. One major approach looks at the type of genome that viruses carry, whether it is DNA or RNA, and whether it is single or double stranded. Another approach focuses on capsid structure, which can be icosahedral, helical, or more complex. These structural differences are not just cosmetic, they influence how viruses enter cells, how stable they are in the environment, and how they spread between hosts. Another important way we classify viruses is by looking closely at their genetic material and how it functions inside the host cell. Some viruses carry DNA, others RNA, and that material can be single or double stranded. For RNA viruses, orientation matters a lot. Positive sense RNA can be translated immediately by the host cell, while negative sense RNA must first be copied into a usable form. These differences determine whether the virus needs to bring in its own enzymes and how quickly it can begin to replicate. By focusing on genome structure, the virus core, scientists can better predict how viruses behave and once it enters the cell. Capsid structure is one of the easiest ways to visually categorize viruses, but it also tells us a lot about how they function. Icosahedral viruses tend to be compact and stable, while helical viruses wrap their genetic materials in a spiral or a protein. Complex viruses like bacteriophages use specialized structures to infect their hosts. Another key distinction is whether a virus is naked or enveloped. Enveloped viruses are more sensitive to heat, drying, or disinfectants because their lipid envelope is fragile. The five capsid structures represents the main architectural strategies viruses use. Naked icosahedral viruses rely entirely on their protein shells and protection, which often makes them more durable outside of hosts. Envelope icosahedral and helical viruses add a lipid envelope, which helps cells entry but makes them more fragile in the environment. Naked helical viruses wrap their genome in a spiral of proteins without an envelope, while complex viruses like bacteriophages combine multiple structural elements for very specific infection mechanisms. The Baltimore classification system shifts the focus from structure to function. Instead of asking what a virus looks like, it asks how the virus makes mRNA once it's inside a host cell. Since all viruses ultimately need to make proteins, this system groups them based on how they get there. By organizing viruses into 7 classes, the Baltimore system helps scientists predict replication strategies and identify targets for antiviral drugs. It is especially useful when studying unfamiliar or newly emerged viruses. When viruses replicate inside cells, they do not do so quietly. The process often causes major biochemical or structural changes known as cytopathic effects. These changes can disrupt normal cell function or destroy the cell entirely. Interesting, many symptoms we associate with viral infections are not caused directly by the virus, but by the immune system responding to the infections. Fevers, inflammation, and fatigue are signs that the body is trying to control and eliminate the virus. The overall impact of infections depends on how aggressive the virus is and how effectively the immune system responds. Viruses also differ in how they leave infected cells. Some cause lysis, where the cell bursts and releases a large number of new virions all at once. Others trigger apoptosis, which is a controlled form of cell death that limits damage to nearby cells. Envelope viruses often leave by budding, where individual virions pinch off from the cell's membrane. In this case, the cell is not immediately killed but is usually damaged and unable to function normally. Although viruses differ in structure and genome type, most follow the same basic infection pattern. First, the virus attaches to the specific receptors on the host cell. Once attached, it enters the cell by either injection, endocytosis, or membrane fusion. Inside the cell, the virus replicates its genome and produces viral proteins using host machinery. These components are then assembled into new virions, which are released to infect individual cells. The details change depending on the virus, but this overall sequence helps us understand where antiviral drugs and immune defenses can intervene. Bacteriophages gives us a clear example of two different injection strategies. In the lytic cycle, the virus quickly takes over the bacterial cell, produces many new virions, and then lyses out of the cell to release them. In the lysogenic cycle, the viral genome integrates into the host DNA and becomes a prophage. The cell continues to live and divide, copying the viral DNA along on its own. Under certain conditions, the virus can switch from lysogenic cycle to lytic cycle. This ability to hide inside the host genome is a powerful survival strategy. Non-enveloped animal viruses rely entirely on their capsid protein to enter host cells. Once the virus binds to the receptor, the cell may engulf it through receptor-mediated endocytosis, forming a vesicle around the virus. In some cases, binding triggers structural changes in the capsid and allows the viral genome to pass directly into the cytoplasm. Because these viruses lack a fragile envelope, they are often more resistant to environmental stresses and disinfectants. Their entry strategies are simple but actually very effective. Envelope viruses add an extra layer of complexity to the cell's entry. Like non-envelope viruses, they can enter through receptor-mediated endocytosis. What makes them unique is membrane fusion. Viral fusion proteins allow the viral envelope to merge directly with the host cell's membrane, releasing the viral content straight into the cytoplasm. This strategy is used by viruses like HIV to give them a more efficient way to infect cells. The trade-off is that the envelopes are fragile, making these viruses more sensitive to drying, heat, and disinfectants. Not all viral infections look the same clinically. Acute infections come on quickly, symptoms worsen over a short period, and the immune system eventually clears the virus. Chronic infections are different. The virus remains in the body for long periods, sometimes for life. The continuous or intermittent replication. Asymptomatic infections are especially important from a public health perspective because individuals feel fine but can still spread the virus. Understanding these patterns helps explain some viral infections and diseases are easy to control while others persist in the populations. Latent viruses take persistence to another level. Instead of continuously replicating, they remain dormant within the host cell, sometimes for decades. During latency, no new virus particles are produced and the host shows no symptoms. However, when the immune system is weakened or the body experiences stress, these viruses can reactivate. This leads to intermittent symptoms as seen in viruses like shingles. Latency allows viruses to evade immune detection and remain in the host for long periods of time. Some viruses do more than cause temporary illness. Oncogenic viruses can interfere with the normal controls that regulate cell division. They may introduce genes that push cells to divide continuously or disable genes that normally stop division when something goes wrong. Over time, this loss of regulation can lead to cancer. HPV is a well known example, linked to cervical and other types of cancers. The key idea is that cancer caused by viruses is not immediate. It develops after long term disruption of the cell cycle. Human viruses are incredibly diverse in how they spread and the damage they cause. Some move easily through the respiratory droplets, while others require close contact or exposure to bodily fluids. Many viruses show tissue specificity, meaning they infect particular cell types such as respiratory epithelium, liver cells, or immune cells. Disease's severity can range from mild symptoms to life threatening illnesses, depending on the virus and the individual's immune response. Understanding these patterns helps guide prevention strategies like vaccines and public health measures. Plant viruses face a unique challenge because plants have a rigid cell wall. This means viruses usually cannot enter unless the cell has already been damaged. The damage often comes from insects feeding on plants, weathering events, or human activities like pruning and farming. Once inside, plant viruses can spread through sap, pollen, or living vectors like insects and nematodes. When viruses spread between plants, it's called horizontal transmission. When infected plants pass viruses directly on to their offspring, that is known as vertical transmission. These infections can have serious consequences for agriculture and food production. Unlike animals, plants cannot feel sick, but they do show infections in very visible ways. Viral infections often disrupt photosynthesis and normal cell metabolism, leading to discoloration in mosaic patterns on leaves. Growth may slow, and leaves or fruits can become deformed. While these symptoms may not kill the plant immediately, they often reduce crop yield and quality. That's why plant viruses are a major concern for agricultural and food security. Vaccines is the most effective tool we have for preventing viral diseases. The goal of vaccines is to prepare the immune system before exposure to the body can respond quickly if the virus appears. Some vaccines use weakened live viruses, while others use killed viruses or only specific pieces of the virus. Killed and subunit vaccines are especially safe because they cannot cause disease. By reducing the number of susceptible individuals in the population, vaccines help breaks protect vulnerable populations and people who may not be able to. Live vaccines work by using a weakened version of the virus that can still replicate but does not cause serious disease. Because the virus behaves similarly to natural injection, these vaccines tend to produce strong and long lasting immune responses. Scientists create these vaccines by growing viruses under conditions that make them less effective in human cells. There is a small but real risk that viruses could mutate back into more harmful forms, which is why live vaccines are not recommended for people with weakened immune systems. Not all vaccines need to be updated regularly, but influenza is a special case. The flu virus mutates at a high rate, especially in the genes that code for its surface proteins. These changes can make last year's immune defense less effective. Each year, scientists track circulating flu strains around the world and predict which ones are the most likely to spread. The flu vaccine is then updated to match those strains as closely as possible. This is why getting the flu shot every year is important, even if you are vaccinated before, as part of a treatment. The idea is not to cure the infection directly, but to strengthen the immune response while the virus is already present. By boosting immunity, the body may be better able to control the infection and limit damage. This approach is not used for all viral diseases, but it highlights how vaccines are not just preventative tools, but can also play a role in managing active infections under specific circumstances. Antiviral drugs work differently from antibiotics. Instead of killing the virus outright, they interfere with specific steps in the viral life cycle. These drugs are designed to target proteins made by the virus, not the host cell, which helps limit side effects. By blocking replication, antivirals reduce viral load and give the immune system time to clear out the infection. Their effectiveness often depends on how early treatment begins and how quickly the virus replicates. Antivirals are powerful but require precise targeting. Tamiflu is a classic example of how antiviral drugs work. Influenza viruses rely on an enzyme called neuroamidase to detect and detach from the infected cells after replication. This enzyme cuts the connection between the viral protein and the host cell surface. Tamiflu blocks the neuroamidase, trapping newly formed viruses at the surface of the infected cells. As a result, the virus has a much harder time spreading to the new cells. When taken early, Tamiflu can shorten illnesses and reduce the severity of flu symptoms. HIV is especially challenging because it integrates into the host genome and mutates rapidly. To manage this, treatment relies on combination therapies, meaning multiple drugs are used at the same time. Each drug targets a different step in the HIV replication cycle, such as entry in the cell, conversion of RNA into DNA, integration into the host genome, or mutations of viral proteins. By attacking the virus at multiple points, these therapies reduce viral load and slow the disease progression. Combination therapies also lower the chances that the virus will evolve resistance to treatment. Prions completely break the usual rules of infectious agents. They contain no genetic material at all. Instead, they are misfolded proteins that cause normal proteins in the brain to misfold as well. Over time, these abnormal proteins accumulate and damage nervous tissue, leading to severe neurodegenerative diseases. Because prions do not rely on replication or transcription, they are incredibly difficult to destroy. Their existence challenges how we define infections and reminds us that not all biological threats follow traditional patterns. Viroids are even simpler than viruses. They are made up of only small pieces of single stranded RNA and have no capsid or envelope at all. Like viruses, they can only replicate inside a host cell, but unlike viruses, they do not produce any proteins. Despite the simplicity, viroids can severely disrupt plant metabolism and development. Their effects can reduce crop yield and quality, making them a major concern in agriculture. To wrap everything up, together viruses are not cells. They are incredibly effective at spreading and evolving because they exploit host biology. We classify them based on their genetic materials, structure, and replication strategies, all of which help predict how they behave. Viral infections affect bacteria, plants, animals, and humans, often causing damage directly through immune responses. Vaccines and antiviral drugs remain our best tools for preventing and treatment. Finally, prions and viroids remind us that the infectious agents do not always fit neatly into the definitions of life. When you think about this question, start by remembering what makes HIV different from many other viruses we talked about. Think about how quickly it replicates and how often it makes mistakes when copying its genome. Those mutations matter. If a treatment targets only one step in the viral life cycle, what happens when the virus changes just enough to get around it? Now shift your attention to the replication cycle. HIV does not rely on a single process to reproduce. It enters the host cell, converts RNA into DNA, inserts that DNA into the host genome, and then produces and processes viral proteins. Each of those steps requires specific viral enzymes. Ask yourself, What would happen if you block just one of those steps versus blocking several all at once? Finally, think about this from the evolutionary perspective. HIV populations inside the body are huge and diverse. If one viral particle survives a treatment, it can help the infection going. Using multiple drugs at the same time changes the odds, making it much harder for the virus to adapt and continue replicating. This question is really about understanding viral evolution, mutation, and how treatment strategies are designed to stay one step ahead.